Thank you. I'm really excited to be here speaking today at the DNAQUAnet conference, coming to you from Georgia. I'm going to talk today about marine sediments, and this talk is really going to be focused on eukaryotic metabarcoding studies. Just to give you a little bit of background about what my lab does, we use marine sediments as this really neat case study for big questions in ecology and evolutionary biology. So most people look at this picture and are interested in the octopus. I look at this picture and I'm interested in the background. So these vast featureless habitats that underlay 70% of the Earth's surface. So wherever there's an ocean, there's marine sediments. And these habitats are really difficult to sample. For the most part because they're so deep and remote. And so marine sediments are very poorly characterized. We haven't even mapped most of the ocean bottom, never mind characterize the species that are there. And the majority of biodiversity in marine sediments is also microbial. So both microbial in terms of prokaryotes, but also microbial eukaryotes. And my lab focuses on what's known as myofauna. So these are microbial metazoa, which are usually about 45 microns to a millimeter in length. And these are species with extremely high biodiversity in marine environments. We're not exactly sure why, because of the obvious lack of biogeographic barriers. But we know there are a lot of species, and they're also very limited in dispersal. So for these reasons, and as well as because of the taxonomic deficit, which is just immense, we have this huge knowledge gap in our understanding of global eukaryotic biodiversity. So that's about the last pretty picture. Unfortunately, I'm going to show in this talk, I'm going to show a lot of graphs. And I really want to use our work on marine sediments to talk about some of the ongoing challenges that we face in the current bioinformatics landscape. So there's a lot of, there's a lot of challenges with data analysis, but some of these issues are extremely pronounced for studies of microbial eukaryotes, such as metazoa. And by metazoa, I'm generally talking about nematode worms because that's my taxonomic background, but this also applies to studies of copepods and tardigrades and about 20 different small animal phyla. So the first challenge which the entire field of microbial ecology is facing is this recent and very significant shift in the way that we cluster things from metabarcoding datasets. So we used to use operational taxonomic units clustered at a sequence identity level of 97 to 99%. And these were pipelines such as usearch or uclust. But now we've really shifted towards delimitation of amplicon sequence variants. So now we have new pipelines such as DNAQUAnet and deblur. And ASVs are exact sequence variants. So they're essentially 100% OTUs, or another way to think of them is just dereplicated sequence reads with some error correction models that are specific to Illumina data. And I'm going to use both OTUs and ASVs in this talk, but I'm also going to use the term MOTUS, which is a more general term to refer to any sort of operational taxonomic unit that's derived from molecular data sets, including metabarcoding data. The second challenge that is really pronounced for eukaryotes, especially small eukaryotes, is that the pipelines that we are using on a daily basis for data analysis are not designed for our needs. So these pipelines are really optimized for the most part for analysis of prokaryotic taxa, so 16S ribosomal RNA genes if you're looking at bacteria or archaea. But also there are other pipelines, other databases, other tools which have been optimized for much larger eukaryotes. So I'm thinking about cytochrome C oxidase pipelines for vertebrates, things like fish. And, small worms are neither fish nor bacteria. They're in the middle. They have eukaryotic biology, but in some respects they are, they do function a little bit like bacteria in terms of the scale that we're thinking at. So the parameters and the specific computational workflows don't necessarily take into account the unique biology that we're working with. And then the third challenge that everyone is facing is reference databases. I'm not going to spend too much time talking about this. I just don't have the time in this talk, but I wanted to highlight that for all of us, regardless of the loci or the taxon of interest, the general sparsity of reference databases is really impacting the accuracy and the performance of our taxonomy assignment pipelines. So this is after the ASV generation step, where you're trying to figure out what your sequence is and match this unknown nucleotide fragment to a species name. I'll talk about it a little, but that's just something that everyone's facing, even if you work on fish. So for this talk, I'm going to show some results from some recent benchmarking tests and evaluations of bioinformatics pipelines that we've been doing, really to evaluate the shift from OTUs to ASVs. And we've been focusing on two different types of data sets, kind of centered around the idea of complexity. But I also want to highlight in this talk that I'm really going to be focusing on patterns and considerations that we've seen in 18S ribosomal RNA, so the nuclear genetic locus that we use for microbial eukaryote studies. So going back to this idea of complexity, we've been thinking about metabarcoding data sets kind of in across a spectrum of complexity. The first and probably the most common to a lot of people watching this talk is a high complexity metabarcoding data set. So this is the most common scenario where you're just doing whole community metabarcoding. So in our case, we're doing metabarcoding of Arctic sediments for one project in the lab. We are trying to get rid of a little bit of the sediment, so there is some physical manipulation going on. We tend to use a 45 micron sieve to concentrate the nematodes and the other microbial myofauna on the sieve. So if there's a little bit of concentration, but not much, then we basically go into DNA extraction from there. But on the other end of the spectrum, one of the things that we've started to do in the lab is actually to try to physically reduce the complexity of these data sets. So instead of just throwing everything into a tube at once, we've added a really labor-intensive microscopy approach to actually look at individual animal specimens, in our case nematode worms, to really capture both the biology of the worm itself, but also to capture the holobionts. So things like symbionts or parasites or any types of gut contents that might be in the gut tract of the worm. So this is really leveraging the fact that we can do taxonomy on these animals. It's not fun, it takes a long time, but we can do it. And having that microscopy approach coupled to the metabarcoding has yielded some really useful insights into these types of data sets. And just to give you a general workflow on how we do this in the lab, so there are microscopes involved. We are generally picking nematodes like we would if it was a taxonomic study in a petri dish under a dissecting microscope. Then we mount them onto slides, identifying them under high power. And then after that, we can remove them from the slides and put them into tubes. So we're taking an identified nematode, putting one worm into a DNA extraction, and then individually indexing those worms in tubes. So when we get the metabarcoding data set, the barcodes, the samples are affiliated with a single worm. And we are, for the most part, we're focusing on the V1, V2 region of 18S ribosomal RNA. Just that end of the gene tends to work better at separating species for nematodes. The benefit of this, and really the strong selling point, is that each nematode specimen ends up being visually identified. So each metabarcoding library, we know exactly when went in that tube. We have the taxonomic information, and we also have video capture images. So we are generating a permanent voucher that's affiliated with that metabarcoding data set, which is important because our sampling is actually destructive. So this morphological voucher can be used to look at things like the gut contents. It can be used to look at the nematode taxonomy itself, because sometimes we end up correcting the taxonomy based on the sequencing data. And another thing we've tried to do as well is to generate a full-length Sanger barcode for the 18S ribosomal RNA gene. So getting the entire 1600 base pair gene fragment in 3 separate chunks and depositing that into GenBank 2 and trying to build up the reference databases. So this takes a long time. It can be expensive, but it's satisfying in a way where community metabarcoding isn't, because you lessen the complexity of your data set, and you can look at patterns affiliated with different genre, different families, and you can start to build a better picture of phylogenetic patterns or microbial associates affiliated with worms. So I'm going to focus a little bit on the technical side of things today, but there's a lot of interesting biology that I again don't have time to put in this talk that I'm happy to answer as part of the question and answer afterwards. So the first thing that we wanted to use these two data sets to look at was, how does this transition from OTUs to ASVs impact the way that we're looking at data? So we've been carrying out a series of tests with both of these data sets and evaluating the 4 major bioinformatics pipelines that people have been using to analyze these data sets in the past and now. And the 4 methods I'm going to talk about in the upper right hand, you can see. These will be consistent across my slides, and the coloring should be consistent too. So first we have euclust, which was the default algorithm in QIIME 1. Then we have vsearch. So vsearch is the replacement for euclust in QIIME 2, but it works and from a similar, it's a similar algorithm. It's just a little bit different. Then we have our ASV algorithm. So we have DADA2, which is generates ASVs based on an entire Illumina run. So you have error correction happening on a whole data set all at once. And then deblur is the last pipeline, which is kind of an alternative to DADA2, but deblur functions, it's really optimized for computational speed, and it does its error correction at the level of the sample. So it's not analyzing the entire Illumina run at once. It's actually looking at sample by sample and doing the error correction that way. So these are slight but very significant differences in the way those last 2 pipelines function. So we've taken these high complexity data sets and low complexity data sets, and we've analyzed them in all 4 pipelines. And I'll go through some of the results that we're seeing. So the first obvious thing we wanted to look at was the number of process reads per sample. On the whole, unsurprisingly, ASV pipelines recover less reads versus OTU pipelines. So ASV pipelines tend to do error correction on the raw FASTQ files that come from the Illumina machine. And they have a stringent error correction model that they're applying to the data sets. So that's kind of in opposition to OTU pipelines, where you tend to demultiplex and do your error correction upstream, which is a little bit less rigorous. Deblur returns slightly less reads, but on the whole, there's not really a huge difference in the total number of process reads across the 4 pipelines. Next, we wanted to look at MOTUs per sample, so the number of clustered units or sequence kind of representative sequences that come out of the pipelines. And here's where we start to see significant differences. So OTU picking pipelines, euclust and vsearch, return 10 times more OTUs compared to ASV generation pipelines, which again is probably unsurprising given how you've changed the way that you're clustering or determining representative sequences for the data. We do have some slight differences you'll notice in DADA2 and deblur. So here's where complexity kind of starts to come in. For example, in the high complexity Arctic sediment data set, where we're doing community metabarcoding, you actually get more sequence reads, more MOTUs per sample in deblur versus DADA2. But when you look at the overall data set, you actually end up getting less overall MOTUs in the entire data set, which I'll show in a few slides. And that situation is reversed in the low complexity data set, where you get fewer MOTUs per sample in deblur versus DADA2. So the green and blue samples in those, the green and blue colors in those, the box plots. Moving on to a little bit more of an overview of the data. So if we plot the number of reads against the ASVs and OTUs, generally what we see is an upward trend. So the more reads that you have per sample, the more OTUs that you tend to recover in your data sets. And this is consistent across all of the 4 pipelines. But again, we're seeing the OTU picking pipelines, euclust and vsearch in this figure are returning 10 times as many more OTUs. So those lines are much higher above DADA2 and deblur. But the complexity again plays a role here. So we start to see impacts in the biology that we're recovering across ASV pipelines. For example, on the right-hand side here, for the low complexity data set where we're metabarcoding single nematodes, de Blur is actually kind of, the curve for de Blur here is flattening out. So even though you're increasing the number of sequence reads affiliated with a single worm, you're not necessarily recovering more OTUs per sample. So that blue line here is pretty flat for the single nematodes. And again, I will talk a little bit more about that on the next few slides. When we summarize the data set, so if we wanted to look at the total number of MOTUs, so not looking at per sample statistics, but this is the entire data set, here's where the real differences start to come in. So the first thing that we see is the really giant difference in the number of MOTUs between the OTU picking pipelines and the ASV pipelines. So DADA2 de Blur returning many thousand fewer OTUs regardless of the dataset complexity. And surprisingly, actually, UCLUS, so the older algorithm from QIIME 1, gives you fewer OTUs compared to the newer OTU picking pipeline in QIIME 2. And that's really because UCLUS is a heuristic search. It's not exhausting search space. It's designed to be fast. Whereas vSearch is generating all of the pairwise alignments using the Needleman-Wunsch algorithm. And that's what we think is related to those differences. So vSearch is actually increasing your amount of OTUs. But then, we wanted to look at this data set in a little bit more depth, and we thought that a good thing to do would be to look at the percentage of this data set, which actually represents the rare reads. So things which may not be biologically valid or may be biologically valid, but getting into the lower read depth of the MOTUs. And here's where the patterns become really stark. So what we see is that the older OTU picking pipelines, UCLUS and vSearch, the vast majority of their data sets, anywhere from 70% up to 96.5%, in these left two columns here, those are MOTUs with under 10 reads. So you're talking about, tens of thousands of MOTUs with under 10 reads. When you look at the ASV pipelines for the same data set, most of that goes away. So your sequences under 10 reads really drop to like a fraction of that in DADA2 and de Blur. But I wanted to note here that de Blur is pretty aggressive in getting rid of these rare MOTUs, especially in complex data sets. So the higher the complexity of the data set, the less rare reads you actually recover, versus the low complexity data set on the bottom, where you have a higher percentage of those MOTUs under 10 reads. And we can look at this in another way. We can look at this in as rank abundance curves of MOTUs. And so here's all the rank abundance curves for both data sets across all 4 pipelines. And we see a pretty significant shift in the shape here. So OTU pipelines tend to return an L-shaped rank abundance curve. And the bottom of that L is basically all of those rare OTUs that are below 10 reads per OTU. On the flip side, the ASV pipelines, because they're aggressively eliminating most of those rare sequences, you tend to see more of an S-shaped curve. So the curve difference varies. You'll see a little bit between DADA2 and de Blur. DADA2 has a little bit more of a long tail, and that's again related to this aggressive elimination of the rare OTUs in the de Blur pipeline. If we wanted to associate with single nematode specimens, so we can visualize this in another way. So these are individual MOTU profiles of individual nematodes, and the samples are labeled with the shapes. What we tend to see is typical with any sort of eukaryotic metabarcoding data sets is that the dominant sequence, what we call the head, tends to be an order of magnitude more abundant than the second-most ASV. And this dominant ASV tends to correlate with the Sanger barcode. So if we were to do old school PCR and Sanger sequencing, that ASV will correspond to the full-length 18S barcode for that species. But then, in addition to that dominant MOTU, we also see this tail of ASVs with low read counts. And this doesn't entirely go away when we shift to ASV pipelines. It becomes less long, so it becomes a shorter tail, but the tail is still there. So these, because this tail of rare reads and rare MOTUs is still consistent in ASV pipelines, we think it's biologically valid, and it can provide really important insights into these individual species. So this head-tail pattern is pretty typical for eukaryotes. The good thing is that the most abundant MOTUs, so this head MOTU, which represents our species barcode, it's really resilient. So even if you're using an older pipeline or vSearch, a newer, older OTU pipeline, your most abundant OTUs are most likely biologically valid. So we can consistently, we've mapped the nucleotide sequences across pipelines, and the most abundant reads, the most abundant representative sequences are always recovered across pipelines, which is a good thing. So the majority of the variation seems to be in this long tail of rare sequences. Looking at this another way, so zeroing in on the low complexity nematode data set, this is where some of the differences in the questions and the hypotheses start to crop up. So we've been looking at the shape of these S-curves for the ASV pipelines to really try and interrogate the biology a little bit more. And the flatter the curve, there seems to be some complex and interesting biology happening here. One example that I'll highlight is from the family Oxostominidae. So these are kind of featureless worms. They tend to be long and pointy at both ends with a whip-like tail. If we plot the single worm ASV profile according to the relative abundance of each of these ASVs, you can see there's some pretty interesting things going on here. So we seem to have many more variant ribosomal RNA genes here. So we have a head OTU with 38% abundance, but then we have at least 3 other OTUs which range from 10 to 17% abundance. So preserving some sort of genomic variation that seems to exist in this species. We don't really know why. This has been pretty well documented in nematodes at this point. We think it may relate to life history and population level factors. The next step will be to generate whole genome sequences and sort of ask this in more of a metagenomic context. But at least the metabarcoding datasets provide some solid ground for hypothesis generation. And then another scenario that we've come across is really not nematode biology, but things that seem to be associated with nematodes. So this is a single worm ASV profile for another species, Daptonema, which again has pretty simplistic morphological features. But here we're finding more on the association side of things that are interesting. So 52% of reads associated with the single worm metabarcoding profile seem to be affiliated with a fungal taxa. And there's been some pretty well documented fungal associations with marine nematodes in the past, observational or anecdotal evidence, but it's exciting to see this recovered in metabarcoding dataset too. So this long tail of rare sequences is important for both the target specimen as well as all of the associates that may be living in conjunction with this specimen. Of course, we don't really know where this DNA has come from. It could be extracellular DNA, it could be gut contents, but it could very well be symbionts or associates. And again, other sequencing approaches or other experimental approaches can be used, in following these observations in metabarcoding datasets. And we can zoom out and look at this across families. So this is the same sort of graph, just looking at the phylogeny of 4 different nematode families. And what we're finding is basically the signal across nematode families and the influence of this long tail seems to differ a little bit. So again, not sure why. Some nematodes seem to have a lot of associates. Some nematodes seem to have barely none. But the only way that we would ever know this was to use this low complexity approach where we're metabarcoding these individual nematodes. We never would have come up with this from a whole community approach. So with that, I'll finish up, and I just wanted to kind of highlight some of our findings. So if you're using older OTU picking algorithms, I wouldn't worry too much. We know they inflate diversity, especially for rare taxa and this long tail. But ASV pipelines aren't necessarily better. They error correct more and they cut down that long tail. But your dominant, your abundant OTUs are still trustworthy. They still correlate with the abundant ASVs, which is the equivalent of your species-specific barcode. And then what I would say is we found that DADA2 is performing really well. That's what we're recommending for eukaryotic metabarcoding studies. It tends to preserve the biology a little bit better. And that's in contrast to DeBlur, which is taking these computational shortcuts for speed, and it's performing this per sample error correction, but it seems to be erasing a lot of the interesting biology that's cropping up in this long tail of rare taxa. And then finally, all of this would not be possible with reference barcode databases. Some of the observations and the issues we may be seeing may be related to the poor number of sequences, the eukaryotic sequences in online databases. We're not quite sure. But again, the reference databases are going to be critical to maximizing this kind of approach. So with that, I'll just say thank you to my lab members and my collaborators and my funding agencies, which have facilitated this work, and I will happily take any questions. Thank you.